Hello, welcome everyone to the workshop in Pathogenesis of CF Airways Infection, NACFC 2021. I am your chair, Dr. Sebastian Riquelme from Columbia University in New York. And with me is our co-chair, Matthew J. Wargo, Doctor, Associate Professor at the University of Vermont Larger College of Medicine. Dr. Wargo is behind the scenes right now, handling all the questions that you may have during the presentations. And we encourage you to participate and to put your question on the Slido window next to the screen where the speaker is talking. So please participate. Before moving forward, I would like to make an announcement about the final of the Junior Investigators in Basic Science Award, which will be celebrated on November 17 between 11 a.m. and noon 30. If you want to have access to the link, please feel free to contact me or contact Karen Tagle from the Cystic Fibrosis Foundation, and we'll be happy sharing this information with you. So before moving forward, what will we discuss today in this workshop? We will discuss the molecular factors that determine the susceptibility to infection in the CF lung, particularly understanding the airway components contributing beyond mucus accumulation, including pathogen interactions with other microbes and with the regionally and temporally heterogeneous inflammation in the CF lung. Also, we will describe how altered host lung defenses provide a specific metabolic advantage to inhale microbes and alter their interactions, focusing specifically on how these microorganisms interact, infect, and persist in the oxidizing environment of the CF airway. So for this, we have 5 presenters. We have, the first one is Alison Weld. She's a graduate student from the University of Pittsburgh. And Alison will describe the interaction between viral pathogens and epithelial cells in the CF lung, and how this influences the ability of Pseudomonas aeruginosa to persist in the respiratory mucosa. Unfortunately, Alison is not here today, but on her behalf, at the end of the presentation, Dr. Jennifer Baumberger, who is part of this study, will be answering the question. So please, Paul, let's see the video.
I'd like to thank the organizers for the opportunity to present to you today. I'm a graduate student in Jem Baumberger's lab at the University of Pittsburgh. I'm going to tell you a little bit about my work looking at Pseudomonas aeruginosa and viral co-infection. I have no relationships to disclose for this presentation today. So as many of you know, chronic respiratory infections are the leading cause of death in people with CF. From many years of observations, we can see clear trends in the microbiology of the respiratory tract as individuals age. So Pseudomonas aeruginosa increasingly becomes the leading pathogen causing infection in adults. And Pseudomonas infection is associated with declining lung function, low microbial community diversity, and worsening morbidity and mortality. So loss of microbial diversity is a hallmark of many chronic inflammatory diseases and correlates with disease severity. In CF, the greatest bacterial diversity is in younger patients with milder disease and good pulmonary status. So as individuals age, they begin to lose diversity, richness, which is the total number of species, and evenness, which is the abundance of each species. However, as patients age, you also see an increasing prevalence of Pseudomonas and Burkholderia, which tend to dominate when they're present in a community. So in work from previous studies, we can see that patients with higher pulmonary function, shown here as FEV1, do have higher community richness, suggesting that bacterial diversity is really important predictors of lung function. So we want to understand what contributes to microbial community shifts. Specifically, we want to know if pulmonary exacerbations might play a role. And these are periods of acute worsening of respiratory symptoms and occur periodically throughout an individual's life. So what are the causes of these exacerbations? About 40 to 60% of pulmonary exacerbations are associated with acute respiratory viral infections. And the 3 main causes of these are influenza, human rhinovirus, and respiratory syncytial virus, or RSV. So there's a wealth of clinical literature to suggest that Pseudomonas infections correlate with acute viral infections. We see first acquisition of Pseudomonas infection within respiratory virus seasons. And specifically, of all of these 3, RSV infection alone was most associated with acquisition and development of chronic Pseudomonas infection. So looking back at this graph of community dynamics, could repeated viral infections actually be driving this change in Pseudomonas dominance that we see over time? So our lab wanted to understand how RSV impacted Pseudomonas virulence. So we first looked at its effect on biofilm formation. So you see that RSV co-infection does enhance Pseudomonas biofilm growth. And so we look here at our epithelial co-culture biofilm model, where we take CF airway cells and perfuse media over the top, so that Pseudomonas has to stick and form a biofilm or will be washed away with the media. So with the preceding respiratory syncytial virus infection, you see increased aggregates here in green of Pseudomonas. So to summarize our previous work, we found that RSV infection triggers an immune response that leads to production of interferon and interferon-stimulated genes, or ISGs. And so these increase the trafficking of iron-bound transferrin to the apical surface of the airway. So when we measure transferrin and apical cell secretions via Western blot, we can see a significant increase in transferrin abundance, resulting in increased iron concentrations. So Pseudomonas can respond to and use this source of iron. So in the data you see here, you can see that we no longer have a biofilm increase following depletion of transferrin alone from apical secretions from RSV-infected cells. Therefore, we can conclude that transferrin is the host secreted factor that's driving this change in Pseudomonas biofilm formation. Importantly, we've also verified that iron and transferrin are also increased following virus infections in both mouse models and clinical studies in humans. So we want to understand how the host is altering its nutritional environment, and how Pseudomonas is sensing and responding to this change. If this is the same process that might underlie the microbial shifts that we observe with CF individuals' age. So do viral co-infections actually change the microbial community interactions? And so to determine this, we wanted to understand how Pseudomonas was responding to RSV infection. So our lab conducted a dual RNA sequencing study where we infected polarized CF airway cells with Pseudomonas alone or Pseudomonas and RSV. And following incubation, we extracted RNA and compared the Pseudomonas transcriptomes of each of these environments. So there were a number of significantly upregulated and downregulated genes, but there was one operon of genes that was significantly upregulated during RSV co-infection. And these genes encoded toxins secreted by the type 6 secretion system. So we wanted to understand if this could impact bacterial competition and microbial dynamics. So the type 6 secretion system is a unique contractile protein secretion system that resembles a syringe. And this system functions to transport proteins directly to another cell. So type 6 is unique in its ability to target bacterial cells in a process interspecies competition, where it can kill bacteria in a number of ways. But it can also target the host cells in order to evade the immune system, resist clearance, and persist in the host. So Pseudomonas actually has 3 of these systems, although today we're going to specifically be talking about the second one, which has previously been found to be involved in host cell internalization and interbacterial competition. So during RSV infections, the structural genes comprising this H2 type 6 apparatus that are needed to export toxic vectors also display a modest increase in expression. However, the genes we're most interested in are involved in this T-set toxin operon. So this operon encodes 5 genes necessary for toxin delivery. There are 2 chaperones, CoTec-T and Tec-T, that function to deliver the toxic effector T-set to the secretion apparatus. T-set binds to the C-terminal extension of the PAR4 needle tip. And then there's also a T-set immunity protein that functions to neutralize T-set if Pseudomonas gets injected by a sister cell. So the rest of this presentation will focus on these 2 questions we want to use to understand T-set. Why is it expressed during co-infection, and how does it impact interbacterial competition? First, we want to understand why it's expressed during co-infection. And this is a really exciting avenue, as most natural inducers of type 6 secretion systems during infection and in the environment are largely unknown. First, we wanted to understand if multiple viruses could induce T-set or if this was an RSV-specific phenotype. So we see during rhinovirus infection, we also see induction of the T-set operon genes and the structural genes of the H2 system needed to express these genes. So we wanted to know if expression was due to a host-secreted factor or the virus itself. So we collected apical secretions and incubated Pseudomonas directly in these secretions. And indeed, a host-secreted product was responsible for expression, as we got expression of the T-set genes and the structural genes. suggesting Pseudomonas is responding to something that the host is secreting. So to begin to understand what was influencing the expression of the system, we designed a set of fluorescent reporters so we could track the transcription of each of these loci. And we measure this by screening GFP signal in a 96-well plate format. And to verify that these reporters work, we used previous information published previously that this system is also regulated by LASR quorum sensing. So we use an exogenous quorum sensing signal and a quorum sensing inhibitor to verify that this screen did work. So we started by screening a variety of nutrients and carbon sources that we knew were altered during virus infection, or that molecules bacteria might encounter during infection. And so we found many molecules that did not induce expression of the system, shown here. However, we were excited to find that iron, shown here in orange, specifically induced expression of the system, matching our previous observations that Pseudomonas can utilize and respond to changing iron concentrations during viral co-infection. So as we know, transferrin availability specifically drives these changing iron levels. And so we also found that transferrin alone could also stimulate the same response. And you can see here this promoter activity is quantified. And so we wanted to confirm this through another method. So we used QRT-PCR to show that the addition of iron does induce expression of this T-set system. And this expression happens quickly, but also persists over a period of 24 hours. So therefore, altogether, this data shows that changing iron concentrations in response to viral co-infection does indeed impact the expression of Pseudomonas genes that are known to be involved in interspecies competition. So now that we have shown T-set is induced in response to iron and transferrin, we want to understand how T-set impacts interbacterial competition. So we began by testing Pseudomonas competition with E. coli as a model gram-negative species. And we're assessing competition through changes in competitive index, which is the ratio of Pseudomonas to E. coli over time. So in nutrient-rich media, we see that wild-type Pseudomonas outcompetes E. coli quite well. However, if you delete this entire T-set operon, you lose competition. And this is specific to the T-set toxin alone. Then if you inhibit secretion by the H2 type 6 system, you also lose competition. And so we wanted to know if this was also relevant on our same airway model as before. And indeed, we see the same phenotype. However, competition on airway cells is even higher than it was in LB media. So to determine how RSV infection impacts this competition, we competed Pseudomonas and E. coli and the apical cell secretions from mock or RSV infected airway cells. In mock infection, you see the T-set operon is still abrogated in its ability to compete with E. coli. However, Pseudomonas still wins as the competitive index is greater than 1. However, in RSV infection, due to the strong induction of T-set in this environment, the microbial community actually shifts so that E. coli wins in the absence of the T-set toxin. Therefore, Pseudomonas relies on the induction of the specific toxin in this environment to compete with other species. And so from data from the same competition showed previously, we see Pseudomonas growth really does not change much in these conditions. However, E. coli growth is driving this change. So you see E. coli is not killed by the T-set operon mutant in RSV secretions because Pseudomonas cannot express this toxin, leading to an increased amount of E. coli present and actually shifting the balance of this community. And so we wanted to know that if iron alone was sufficient to also drive this competition. So when we add iron to airway cells, we see an increased competition and corresponding defect, which is about 1 log less in the T-set operon mutant, the T-set mutant, and the H2 type 6 secretion system mutant. And if we look at E. coli survival, we see about a log less of E. coli CFUs in competition with wild type in iron-containing environments. So this shows that iron concentrations indeed change Pseudomonas behavior and allow it to better compete with other species. So in summary, we've shown that viral co-infection increases iron availability and leads to enhanced Pseudomonas biofilm formation. And this is due to increased transferrin release. We know that the T-set toxin locus is expressed during multiple respiratory viral infections, and that gene induction is due to host-secreted iron and transferrin. We know that the T-set toxin is active against other bacteria, An increased expression of T-set in addition to iron or during RSV infection actually increases competition and can alter the microbial community dynamics. So in our working model, we know that younger children with CF have much more diverse microbial community and better pulmonary function. But over their life, they experience pulmonary exacerbations and acute viral infections. And we know acute viral infections increase the amount of iron available in the airways. So Pseudomonas can respond to this iron and become more competitive and have increased biofilm formation. So over an individual's life, this might actually drive Pseudomonas dominance, as we see in older patients that have much more Pseudomonas in their airways. And this gives further support for the use of iron chelators as therapeutics, as this might actually help preserve the microbial communities we see in the airway. So I'd like to thank everyone that's helped in this project, including my mentor, Jen Bomberger, and our collaborator, Patrick Thibodeau, along with two former members, Matt Hendricks and Jeff Melvin, who contributed to most of this preliminary data. Thank you for your time.
Okay, thanks very much, Alison, for that presentation. That was brilliant. So we have a couple of questions that we would like to discuss with Dr. Bomberger, who's in behalf of Alison because she's not here today. And so one of the questions that I have out of this presentation is why iron is connected with this type 6 secretion system, the H2 type 6 secretion system. Why not another one? Because Pseudomonas express 3 of them. So what is the real connection with specifically that part of the genome?
Thanks for the question, Sebastian. That's an interesting question. I mean, I guess the why I'm not so sure about. So definitely the type 2 system, the H2 system has a fur box, and so it is regulated by iron. But the T-set operon itself, we don't know that much about actually what nutrients regulate it at this point. And so the why I don't have a good answer to, the teleologically, I'm not actually sure the why of.
Let's say how.
Yes, so that part we are certainly actively looking at. And so we're looking at the promoter now, trying to understand how iron is regulating the T-set operon itself. It's not actually obvious based on the different motifs that are in the operon, in the promoter to the operon. And so that's something that we're still definitely looking at, but don't have a great answer for you there yet.
Okay, no, that's super interesting. And also at this time, I encourage the people to participate, asking their own question in the slide application. And I would like to ask you about what is the role of heme here? Because heme as well is a minor iron source. And this model, it's what is happening, what we are seeing with infection with RSV, it's during an acute infection. So during the chronic infection, based on some reports, it looks like Pseudomonas evolves to consume more heme compared to iron. So can you elaborate a bit more what is going on during that transition and how RSV is regulating that part?
Yeah, that's a great question. So, so Alison's looked at a bunch of different iron sources. And so in the presentation, she showed the data with holotransferrin. We do get a partial phenotype if we give the bacteria hemoglobin as an iron source. But it does seem to be the largest phenotype we get for this induction is really in ferric chloride and then with iron bound to transferrin. So giving the bacteria holotransferrin. And so she's also taken a look at how Pseudomonas is acquiring the iron. And so and kind of hot off the presses data, it appears siderophores seem to be important. We get a much smaller phenotype with Hass and Fu, the systems that would bring in heme-bound iron or heme molecules to get iron for the bacteria. And so we think the phenotype is more geared towards pyoverdin uptake of iron sources. And that might suggest that this maybe is more important earlier during in a patient's life, maybe as that transition's happening. But once Pseudomonas has become dominant in the community and really starts that pathoadaptation process, maybe this system is less important. At this point, we are kind of looking through some of our evolved isolates for other studies in the lab, trying to get a handle on that, but we're still looking at that. Okay, great.
That's super interesting. And we have some questions from the audience. Moving forward, we have a question from Pradeep Singh. He says, Alison, very nice talk and extremely interesting. And he has 2 questions. The first one is, does iron increase type 6 mediated host toxicity?
Yes, so we've looked a lot at host toxicity, and that does not appear to be in our epithelial co-culture model. We do not seem to see any increase in cytotoxicity towards the epithelial cells.
Okay, so, and the second question is, does iron increase type 6 mediated toxicity to CF pathogens other than E. coli?
Yes, great question, Pradeep. So Allison has started to screen through these. And so we started with E. coli because that's where the system was initially looked at. But now Allison screened a bunch of more CF relevant pathogens. And she sees it really is differential. And so type 6 seems to be important, or we see competition with a variety of different pathogens, Haemophilus influenza, Stenotrophomonas, Achromobacter, but only a subset of those are T-set mediated, so this toxin mediated competition. And so Stenotrophomonas and Achromobacter appear to be T-set mediated. And for instance, Haemophilus influenza does not appear to be T-set mediated. And then we see Burkholderia is very complicated because various Burkholderia species can express T-CIT, the immunity protein. Yeah. And so that becomes a little more complicated. And we're doing some studies in collaboration with Von Cooper to try to look at Pseudomonas Burkholderia based on the genomes and predicting whether there's competition there or not. So that's a much more complicated question.
Yeah, I'm aware of that competition with the Burkholderia. There are super interesting work about that. Mm-hmm. So I would like to share with you some other questions from the audience. We have another one that is there a particular cell type in the CF airway that's most commonly infected by viruses? For example, given bronchial versus alveolar epithelial cells.
I guess when we think of where the impacts would be in cystic fibrosis, we would think of more upper respiratory tract or airways as opposed to the alveolar epithelial spaces. Mm-hmm. Certainly in shifting nutrients that would be acting in the really local domains of where we think Pseudomonas is, particularly earlier on in disease before patients get to really end stage lung disease. And so I would think the impacts that we're observing and the shifts that we're seeing in nutrients that impact these communities are probably in the upper airway and the upper respiratory tract and the airways, not the alveolus.
Okay, so based on that, on the interaction of TCIT with other cells and the infection in other cells in the airway, we have this question from Anthony Recuti. He said, nice work. Have you looked at the interaction of TCIT on immune cells like macrophages?
Yes, so we've not actually done those studies. So we've talked about doing that in the lab. It's certainly of interest. We've thought about immune cells more in the different types of nutrient or really iron sources that might be available. And that we thought about the macrophages in thinking about the iron sources that Pseudomonas may see to induce this competition. But we haven't done any studies on like cytotoxicity as type 6 can act towards eukaryotic cells. We saw no effect on epithelial cells, but we haven't done the macrophage studies. And so it's a good question. We haven't done it yet.
Yeah, and coming back to the interaction of Pseudomonas with RSV and other bacteria, people are wondering about staph, because staph is another major pathogen, living there in the airway. So do you have an idea about what is going on with staph reacting to this iron release and competition with type 6?
Yeah, so at least in terms of the H2 system for the type 6 competition system or secretion system in CF or in our model system, the H2 does not appear to be have any role for staph killing. There's some suggestions that there may be roles for the other type 6 secretion systems in Pseudomonas with staph, but in our hands and in our models, the type 2 system, the H2 system does not appear to have an effect on staph.
Okay, so as a final question, because I think that we're running out of time, is there any relationship between the production of energy by Pseudomonas to secrete these factors? Is iron promoting energy production in bacteria and like that, and that's the reason why they are secreting more of these toxic proteins?
Yeah, so I guess we haven't done those exact studies to know that. We certainly normalize all of the promoter activities for growth in the bacteria because there is more growth with iron and in RSV secretions. Exactly. But we haven't done the actual studies to look at energy production. So I don't have an answer for that. Okay,
well, thanks very much, Jennifer. That was a great presentation by Allison, sorry. So now we will move forward to the next presentation, which will be done by Satoko Nakano. She's a research technician at Marsico Lung Institute, UNC Cystic Fibrosis Center. And Satoko will illustrate the molecular pathways balancing the infectivity of SARS-CoV-2, the pathogen that produced the disease COVID-19 in the CF lung, Please, Paul, let's move to the next video.
Hello, my name is Satoko Nakano, and I'm a research technician at the University of North Carolina at Chapel Hill Cystic Fibrosis Center, and I will be presenting on pathways balancing SARS-CoV-2 infectivity and disease severity in CF. I have no relationships to disclose related to this presentation. So a little background. CF patients normally have severe reactions to viral respiratory infections such as RSV and influenza. In clinical settings, people with CF have higher respiratory symptoms, higher rates of hospitalization, and lower pulmonary function. And since COVID-19 is also a respiratory disease, it would be expected that people with CF are at risk for severe illness. However, in the case of SARS-CoV-2 infection, the outcomes of people with CF and the general population are similar, suggesting that people with CF may potentially be somehow protected from SARS-CoV-2 infection. We look at a pro-mucin secretory cytokine IL-1 beta because it is upregulated in CF and upregulates ACE2. And IL-1 beta is highly expressed in the airway when inflammation occurs. In CF airways in particular, due to chronic inflammation, there are high levels of IL-1 beta expression, as you can see in this image of cross-section of lungs stained for IL-1 beta through RNA-scope. You can see these red signals are IL-1 beta, and they're only present in the CF lungs and not in the non-CF lungs. The ACE2 is known to be the receptor for SARS-CoV-2 virus. The virus attaches to the cell, and then TMPRSS2 works with ACE2 to allow viral entry and spread. And in our previous study, we found that IL-1 beta treatment for 7 days upregulates ACE2 in the airway. And this could suggest that IL-1 beta could increase infectivity by increasing ACE2 receptors. But still, the relationship between IL-1 beta and SARS-CoV-2 infectivity is not yet known. And in CF, the airway is primed by IL-1 beta, causing chronic inflammation. So it's important to understand how that affects SARS-CoV-2 infection and how that affects outcomes. In our study, we used supernatant from mucopurulent materials, SMM, from CF airways to treat fully differentiated human bronchial epithelial, or HBE cells, in culture to mimic in vivo conditions. And this is a figure from a paper that was published by the group we obtained our SMM from. And they analyzed the components of their SMM, and you can see that IL-1 beta here is one of the main components of SMM, but there are other cytokines and interferons in the mixture. And because IL-1 beta is one of the main components, we hypothesized that SMM would act similarly to IL-1 beta alone. And we have two major goals for this study. The first one is to test how IL-1 beta affects SARS-CoV-2 infectivity in airway epithelia. And the second one is to understand the mechanism of SARS-CoV-2 infection in inflamed CF airways. So our first finding is that IL-1 beta increases SARS-CoV-2 infectivity while SMM does not. And here's our experimental setup. We treated cultured fully differentiated HBE cells with IL-1 beta and SMM 72 hours prior to infection, and interferon lambda treatment was done 24 hours prior. The SARS-CoV-2 virus that we used to infect these cells contained GFP in it, so we took a live image of the cells at 72 hours post-infection to observe infectivity. And you can see some infection in the vehicle control here. But when you look at IL-1 beta, you see many more cells are infected. And while we expected to see an increase in infectivity because IL-1 beta is known to upregulate ACE2, the receptor for SARS-CoV-2 virus, the amount of GFP signals we saw was very surprising. And because SMM contains high levels of IL-1 beta, we hypothesized that SMM-treated cultures would also have high levels of infection. However, the number of cells infected by SARS-CoV-2 virus in the SMM pre-treated cells looks similar to the vehicle control rather than IL-1 beta. And below here, this is an RNA scope done on the same cells where we look at the cross-section of the cultures from above. The red signals are SARS-CoV-2. And you can see again that vehicle control and SMM have similar levels of infection, and IL-1 beta has many more SARS-CoV-2 signals. And next, we look at the viral titer. These graphs are shown on a log scale, showing the fold change. And you can see on this kinetic graph, the viral titer over 3 days. Viral titers were measured at 24, 48, and 72-hour time points. And I-1 beta has a higher viral titer at all time points, while vehicle and SMM have similar viral titers, and interferon lambda has a very low viral titer, which is expected from interferon treatment. And looking specifically at the 72-hour time point, you can see that I-1 beta pre-treated cells have about a 10-fold higher titer compared to vehicle and SMM. And we used a paired analysis using linear mixed effect models and found that there's a significant difference between vehicle and I-1 beta, but no significant difference between vehicle and SMM, which is consistent with what we've seen so far. So next, we found that acute I-1 beta treatment does not upregulate ACE2. So we first looked at ACE2 receptor because it's the receptor for SARS-CoV-2 virus. And when the Cell paper was published describing the upregulation of ACE2 after 7-day I-1 beta treatment, it was not yet known that there are two different isoforms of ACE2 in the airway. There is a delta ACE2, which is a truncated version that begins at exon 1C, and it may be stimulated by interferons, and it does not have the N-terminus, so it's unable to bind SARS-CoV-2 virus. And the full-length ACE2 that begins at exon 1A or 1B is the true receptor for SARS-CoV-2 virus. And we designed primers to specifically target each isoform. And we found that none of our qPCR data shows upregulation of ACE2. In the ACE2 data set where we used a probe that can detect both isoforms, it looks like there could be an upregulation here, but there's no statistical significance. And when you look at the isoforms separately, neither upregulated, including full-length ACE2, the true receptor for SARS-CoV-2. So this suggests that I-1 beta does not increase infectivity through the upregulation of ACE2, or in acute treatment, there's a pathway independent from ACE2 that's contributing to the increased infectivity. So next, we wanted to investigate cell type specific host innate response to I-1 beta, SMM, and interferon lambda treated cells to see if there are any differences in gene expression that may be contributing to the differences in infectivity. For this experiment, we treated I-1 beta and SMM 72 hours prior to RNA collection and interferon lambda 24 hours prior to collection. Then we ran a 10x single-cell RNA sequence on these samples that are not infected with SARS-CoV-2. And we did this to see if there's any specific differences in gene expression overall, as well as across different cell types, or to see if any new cell type clusters appear with different treatments. Here's a summary of all 50,000 cells that were analyzed, and they were all categorized based on cell type, and these are all the different cell types that were found. And from our single-cell RNA sequence data, we found that I-1 beta and SMM pretreated cells express different genes compared to interferon lambda pretreated cells. So all these genes shown, they're genes that are expressed in interferon lambda or I-1 beta or SMM compared to control. And in the left here, we compare interferon lambda versus I-1 beta. And genes expressed by each condition are not very well correlated. In the red, you can see the genes that are highly expressed by interferon lambda treatment, and in the green, you see the genes that are highly expressed by I-1 beta treatment. And to the right, we see similarly, interferon lambda and SMM also do not have a good correlation in the genes they express, with the red indicating the interferon lambda treatment and blue indicating the genes that are upregulated by SMM treatment. The ones in red are considered ISGs. So in the previous slide, we looked at genes, at all genes expressed. And in this slide, we look specifically at interferon-stimulated genes, or ISGs. And we found that neither acute treatment of I-1 beta nor SMM up or down regulates ISGs. So to the right side here are all of the ISGs, and the colors up here represent the various cell types that were found, and the 4 color blocks here represent each of the different treatments. And orange indicates high expression, and purple indicates low or no expression. Interferon lambda is used as a positive control, and you can see that ISGs are upregulated in all cell types in the interferon lambda pretreated group. But control I1 beta and SMM show similar expressions of all ISGs with no particularly obvious up or down regulation of any ISGs across the cell types. One interesting thing is that a group of cells that express ISGs in the control group, right here, those are ciliated cells, and it seems that it's expressed in all conditions and further upregulated by interferon lambda. And since ciliated cells are often targeted by viruses, including SARS-CoV-2, these genes could be important in viral infection. But we have not yet looked deeply into what those genes are and their functions. But overall, the main takeaway from this data is that I1 beta does not acutely increase SARS-CoV-2 infectivity by downregulating ISGs. So just to summarize a little bit of what we have so far, we have found that acute I1 beta treatment increases infectivity while SMM does not, despite containing high levels of I1 beta. And we've tested 2 hypotheses in acute treatment of I1 beta and found that it does not upregulate full-length ACE2 receptors or downregulate ISGs. And for future experiments, we would like to study I1 beta-induced inflammatory pathways relevant to viral replication, as well as replicate these studies on chronic treatment of I1 beta and SMM. And next, we looked at differentially expressed genes to see differences in gene expression between SMM and I1 beta. Gene expression between I1 beta and SMM pretreated cells are very well correlated, but there are a few genes here that are highly induced in SMM compared to I1 beta that you can see in the red circle. And this is a list of the top 30 differentially expressed genes that are highly induced in SMM versus I1 beta. The highlighted genes are the ones we're particularly interested in because they're known to be related to viral protection. MUC1 and MUC4 are tethered mucins. BPIFA1 and BPIFB1 are innate immune secretory proteins, and IFITM2 and IFITM3 are transmembrane ISGs. So based on the data from the last slide, we can speculate that SMM-induced viral protective innate immune proteins may offset the effect of I1 beta to explain the similar rates of infectivity to control despite containing high levels of I1 beta. We are also working on knocking out SMM-induced innate viral protective genes in HBE cultures in vitro, such as MUC1 and BPIFB1, to observe its effect on infectivity. And I would like to thank all my collaborators and the entire CF Center for their contributions. I would also like to thank CFF and CFRI for funding this study. And thank you for your time.
Excellent. Thanks very much, Satoko, for that great presentation. That was brilliant. So we have a couple of questions from the audience already. The first one is, people are wondering, is SMM similar to sputum?
Yes, so SMM, I guess, is like the supername of the material that's found in the CF airways. So it is similar to sputum, and that's kind of how we mimic like in vivo situation where we kind of try to make the cells think that they're kind of being primed with what CF airways are primed with. Okay,
yeah, that's an important point. Also, I have this question, but also people are wondering, Tracy Bonifila is asking, do you think the reason that AC2 did not increase with SMM is due to the IL-1 receptor A that is also elevated in the SMM products? Maybe the IL-1 beta is being neutralized. What do you think? Could you elaborate on that?
Yeah, so that's a great question. That's actually kind of what we're looking into right now. We're looking at the NF-kappa beta pathway next to see if that's playing a role in like the infectivity of SARS-CoV-2 with IL-1 beta. So we're doing experiments of using IL-1 RA and treating cells with that, and then also treating with IL-1 beta to see if the I1 beta is actually like upregulating that pathway somehow, causing the increase. And so we're trying to block it and see if that increase in infectivity will change. But I don't have the data for that yet, so I can't answer either way. But
well, that is a super important point. So you are doing that in the future. Well, you will answer the question then. So also, did you see how IL-1 beta and SMM affected the pulmonary ionocytes that we see that you have in your datasets?
So we haven't looked specifically at ionocytes yet. We're kind of still working through our single cell data. It's a lot of data to digest still. We're focusing a lot on like ciliated cells in particular too, since they're the ones that are targeted for SARS-CoV-2 infection in human airways. But definitely, I think we'll have to look more deeply into all the different cell types and what cell types are playing what role in the infectivity.
Okay, great. Also, something that you measured, you did a lot of controls about it, is the expression about AC2. So one of your hypotheses that of your initial hypothesis that probably AC2 was being upregulated by IL-1 beta, and that's the reason why you got more infection, and you didn't see any difference in the different isoforms. But also, there are other proteins in the surface of epithelial cells that are important for the entry of SARS-CoV-2, and there is one certain protease that is called TMPRSS2 that activate the spike protein and promote the fusion of the virus with the cell. So one of the questions that we have from the audience, from Jennifer Bartlett, is, did you observe upregulation of the cell surface protease, TMPRSS2, with IL-1 beta stimulation?
Yeah, so that's also a good question that we're kind of looking into now. We did see like recently a paper published that was talking about IL-1 beta upregulating TMPRSS2. We haven't personally looked into that yet, and we don't know if the increase of TMPRSS2 is just TMPRSS2 is able to, without ACE2 is able to upregulate infectivity or not. So we're not exactly sure on that topic yet, but we're definitely looking into kind of the other important proteins that are there for SARS-CoV-2 viral entry.
And in terms of COVID-19, we know that one of the major problems is the cytokine storm produced in the airway during the infection, okay? And you're using IL-1 beta, which is a super pro-inflammatory cytokines produced by macrophage and myeloid cells mainly in the airway. But these cells also are producing other cytokines. For example, IL-6, that is also produced by viral infection. And we know that if you block IL-6 in some SARS-CoV-2 models, now the inflammation is reduced and the infectivity also is reduced. So do you think that IL-6 or other cytokines also are playing a similar role in your model more than IL-1 beta?
Yeah, I think definitely there's multiple like cytokines and multiple things kind of happening during infectivity. So it's definitely not just IL-1 beta particularly increasing infectivity, but I think we're focusing on that just because we saw this huge increase, especially when we looked at like the GFP picture we had earlier. We just saw this big difference that we really were interested in and wanted to dive into what in IL-1 beta in particular is kind of the pathway relating to IL-1 beta is increasing infectivity. Okay,
and that's a super important point, and also how the SMM and IL-1 beta are inducing these different responses in the epithelial cells. Based on that, you share with us different genes, a collection of genes that were upregulated by SMM in comparison with IL-1 beta. And you mentioned that they are protective against infection. So can you elaborate a bit more how these genes are protecting the host and how that is related to cystic fibrosis?
Yeah, so we're not exactly sure what the genes specifically do relating to SARS-CoV-2 yet, since that's what we're kind of looking into now. But we know like genes such as like BPIF-B1 and stuff are important for other viral infection protective in protection from other viral infections. So we're assuming that they could also be playing a role in protecting the airway because they're like tethered mucins more on the surface, so doing something kind of with the surface protecting viral entry is kind of what we're thinking right now. And so yeah, we created like MUC1 knockout cell lines to see if, and treating them with SARS-CoV-2 to see if infectivity might be any different or might increase more in that case. and trying to test what gene is specifically kind of playing this protective role in SMM treatments.
Okay, amazing. So just as a final question, because we have less than 1 minute remaining. So I like the idea that you have that in the SMM, you have different components that can be changing the susceptibility to infection by a specific virus, in this case, SARS-CoV-2. So in the SMM, as in the sputum, you have different cytokines, proteins, but also you have different other components, such as a lot of metabolites that are being released by the cells in the environment, because you have immune cells that are releasing all these components during their activation. Do you think that also there could be a metabolic component, that we know that there is a change in the metabolites are accumulated in the airway of cystic fibrosis patient? Can they be playing a role here?
Yeah, so that's not quite my I guess my specialty. So I'm not really exactly sure what metabolic roles could be playing in protective on this, but there's like multiple things happening here. There's so many different cytokines and interferons in there that we're not really able to like pinpoint one thing that's kind of been playing the protective role. So we're kind of going like one by one, seeing what could be and seeing if any of these are kind of successes in our hypotheses right now. So.
Okay, excellent. Thanks very much, Tatoko. This was a super interesting talk, and thanks very much for your discussion. Okay, so now we will move forward to the next presentation, and we will have Samantha Durfee. She's a graduate student from the University of Washington, and Samantha, she will guide us through the genetic evolution of Pseudomonas aeruginosa in the CF lung, and how the airway damage influenced the adaptation of these pathogens to different regions of the respiratory mucosa. So please, Paul, let's move to the next video.
Hello, everyone. Thank you all for tuning in today. My name is Sam Durfee, and I'm a PhD student in Pradeep Singh's lab at the University of Washington in Seattle. Thank you to the organizers for the invitation to speak today, and I have no disclosures. The work I'm going to tell you about today was inspired by the observation that lung disease is remarkably heterogeneous in CF. To show just one example of that heterogeneity, here I'm showing an explanted lung from a person with CF. As you can see, the upper portion of the lung has a high degree of damage, while the lower portion of the lung has a much lower degree of damage. And this heterogeneity raised two questions for us that I'd like to address today. First, does lung heterogeneity contribute to pathogen diversification early in disease? And then, does lung heterogeneity affect infection outcomes after treatment with elexacaftor, tezacaftor, ivacaftor, which is the newest CFTR modulator? In order to address these two questions, we performed bronchoscopy to sample 5 lung regions in each of 9 adults who had relatively mild disease. Their FEVs ranged from about 60 to 80%. We performed bronchoscopy before and about 1.5 years after patients started ETI. Now, for a little background for question 1. A single Pseudomonas lineage persists throughout the life of a patient and evolves extensive phenotypic and genotypic diversity. One example of phenotypic diversity is rhamnolipid production, shown here. Rhamnolipids are an important virulence factor. On this plate, there are 96 arrayed isolates, and blue colonies indicate that the isolate cannot produce rhamnolipid, while variations on white indicate that the isolate can produce rhamnolipid. Importantly, all of these isolates were collected from one patient at one time point, so this represents coexisting diversity. Additionally, when we sequence 96 Pseudomonas isolates from one patient at one time point, we see anywhere from 300 to 3,000 SNPs, depending on whether the lineage is a hypermutator. However, the mechanisms of this diversification are not yet understood. We hypothesize that geographically isolated evolution could contribute to Pseudomonas diversification in early disease. When I say geographically isolated evolution, what I am proposing is that the Pseudomonas in the upper portion of the lung could be evolving in isolation from, or could be evolving independently of, the Pseudomonas in the lower portion of the lung. Within isolated evolution, there are two potential components that can lead to diversification. First, physical separation alone can cause diversification by enhancing the effects of drift. And if drift generates specialists, the physical separation can shelter those specialists from competition. In addition to physical separation, if local conditions differ, that can create diversification by generating different selective pressures. And to give you an example of how local conditions within a lung might differ, I want to show you the inflammation data that we gathered with this study. Here, we measured neutrophil elastase shown along the y-axis, and then each subject is shown along the x-axis, and each dot represents one region within the subject. In this first subject, you can see that their neutrophil elastase ranges anywhere from about 2 micrograms per mil up to about 50 micrograms per mil. And these marked differences are not unique in this subject. We observed them again, for instance, in subject 3 and subject 4. However, there are notable exceptions, such as subject 5, who had very little difference between their regions. So, how can isolated evolution be detected? We detect it by measuring the number of migrations using a phylogeny. Here, I'm showing you an idealized phylogeny where each dot represents an individual, and importantly, the color of the individual indicates what region they came from. And so red individuals come from region A, while blue individuals come from region B. In this tree, we see that closely related individuals are frequently found in different regions. For instance, up here. In order for two closely related individuals to be found in separate regions, there had to have been a migration. Now, because we see frequent migrations throughout the tree, we can interpret that there's free mixing between the regions due to this migration. At the opposite end of the spectrum, if you have closely related individuals that are always found in the same region, that indicates there's no migration occurring. And so our interpretation is that this is completely geographically isolated. Now, I don't have time to go into the methods here, but we used a statistical test that allowed us to simulate free mixing and then measure isolation by comparing the number of migrations we observed in our populations to the free mixing simulation it generates. We whole genome sequenced up to 96 Pseudomonas isolates per region per subject, and we've analyzed 3 subjects to date. Here, I'm showing you the migration data from subject 1. The red frequency histogram is the number of migrations that were generated by the statistical test when we simulated free mixing. Now, when we look at our actual observed populations, we only see 37 migrations, which is far fewer than we expected with the free mixing. And so we can interpret that there is isolated evolution occurring. When we looked at our second subject, we saw a very similar pattern where we observed far fewer migrations than we expected based on our free mixing simulations. And so again, in this subject, it looks like PA is evolving in isolation. Finally, in the third subject we looked at, we again saw that the number of migrations we observed was less than that expected with free mixing. In all 3 of these subjects, the p-value for these comparisons was less than 10 to the negative 6, which indicates that isolated evolution is occurring. But I also want to highlight that some mixing also occurs, since we never observed a migration rate of 0. Now, these stats are all fine and good, but I do want to show you one example of a phylogenetic tree from this study. And so here I'll be showing you the phylogenetic tree from subject 1. Again, each color is going to indicate the region that the isolate was found in, and I've color-coded the lung up here to indicate which region we had sampled. So here is the tree, and you can see that in some areas, like here, there is clustering based on the location. And so these areas indicate isolated evolution and are probably what's driving our statistical signal. However, as we saw when we counted the migrations, there are also areas of the tree where there's a lot of mixing going on. So Pseudomonas evolves in regional isolation early in disease, but mixing also occurs. We don't yet know what the consequences of this are, but I'd like to speculate with you for a moment, because we think that the combined effect of isolated evolution and limited mixing could affect disease progression. For example, if you have a region with high neutrophil activity and the Pseudomonas evolves in isolation in that region, you could develop neutrophil-resistant variants. Those neutrophil-resistant variants could then migrate to new regions and establish disease in that new region, since they're able to escape immune pressure. Okay, so moving on to the second question I want to address. Does lung heterogeneity affect infection outcomes after treatment with elexacaftor, tezacaftor, ivacaftor? Studies to date indicate that most people who have chronic Pseudomonas infections will remain infected after their CFTR is corrected. And we hypothesize that after CFTR correction, Pseudomonas persists in severely damaged lung regions and is cleared from less damaged regions. I didn't mention this at the beginning, but when we chose lung regions to sample, we based that decision on the degree of damage. We used chest CT scans to pick the 2 regions with the highest damage and the 3 regions with the least damage within each subject. And then we repeated bronchoscopy targeting the same 5 regions a year and a half after patients started their ETI. Now, this is an ongoing study, but I wanted to show you some early data from these subjects. Thus far, we've had 7 of 9 subjects return for their post-ETI bronchoscopy. And if you'd like to ask me about the final 2 subjects, they will have returned by the time you all are viewing this recording. To start sharing the data, I'm first just going to show you a few examples of interesting subjects. Here, I'm showing you subject 5, and their Pseudomonas concentration expressed as CFUs per mil of BAL fluid is along the y-axis. Their pre-ETI concentration is on the left, and their post-ETI concentration is on the right. Additionally, the red squares indicate regions that had high damage, and the yellow circles indicate regions that had low damage. As you can see, it appears that this subject eradicated their Pseudomonas since all 5 regions were PA negative after ETI. This is super exciting for this subject. However, not many of our subjects eradicated. For instance, we have this subject here whose all 5 regions were positive before ETI and remained positive after ETI. However, all 5 regions did decrease in concentration. Then we have this subject here who had a negative region before ETI that converted to positive after ETI, potentially indicating a migration event where Pseudomonas was able to establish disease in a new region, establish infection in a new region. Finally, we have this subject who exhibited very little change in response to ETI treatment. Now, to try and get at our hypothesis, we wanted to measure whether regions with high damage and regions with low damage exhibited similar responses. Here, I'm showing you the proportion of regions that cleared PA after ETI. As you can see, regions with high damage cleared about 1/3 of regions with high damage cleared, and about 1/3 of regions with low damage cleared the infections. And so there's very little and certainly not statistically significant difference between these regions, between regions with high damage and low damage. Finally, we wanted to look at the change in Pseudomonas concentration after ETI. And so I calculated this as log 10 PA CFUs per mil. And so a -2 log change indicates about a 100-fold decrease in Pseudomonas concentration. As you can see, regions with high damage, shown with red, and regions with low damage had very, very similar decreases in PA after ETI. And any difference that you're seeing there was not statistically significant. So in conclusion, many PA-infected regions remained infected after ETI. In response to ETI, as measured by clearance and change in PA density, did not differ between regions with the most and least damage. While we're still waiting on the final 2 subjects, it appears that our hypothesis is probably incorrect. With that, I'd like to acknowledge all of the wonderful people who have put a ton of effort into this project. This has truly been a team effort, and this would not have gotten done without every single one of these people on the screen. So thank you all very much. And then thank you to all the listeners for your attention, and I'm really looking forward to hearing your questions. Thank you.
Okay, thanks very much, Samantha. That was a great talk, and it wake up a lot of motivation to the audience. We have a pile of questions here that we will start immediately asking you. So the first question that we have is a super popular question by Jennifer Bomberger. She said, terrific talk. Do you have any thoughts on potential triggers for migration events?
Oh, wow, that's a very interesting question. Let's see here. I think you could think about things like population density or potentially like infiltration of the immune factors. Because I guess if you think about it as a biofilm aggregate model, I could potentially look at the factors that trigger migration events out of a biofilm and see if we can find correlates with that in the lung environment. But that's a very interesting question. Thank you.
Yeah, I can see. So also we have other questions from the audience. And we have another popular question that is from James Gurney. He's asking, do you think migration events increase, decrease, or stay stable through an infected person's life?
Oh, that's a super interesting question. Yeah. So I think what goes on is that in the beginning, you have when patients are young, I think you're going to have much less differences between the different regions, like phenotypically, so inflammation, nutrition, stuff like that. And so I think the Pseudomonas are also going to be less specifically adapted. And so I think there's probably a higher rate of migration at the beginning of a person's life. Yeah. And then as you establish damage and plugging and things like that, I think you're going to prevent that migration between the different regions. And so I think end stage disease, when you have the most scarring, the most plugging, restriction, and Pseudomonas has had the longest time to adapt to specific regions, I think you're going to see a decrease in the migration rate. And our numbers are small, but there was a 2015 paper that looked specifically at end stage disease. So these were lungs that had been explanted from patients right before transplant. And at least in one of those subjects, they had a much lower rate of migration than what I'm seeing in these kind of middling stage people. But
I see.
That's it.
Yeah, I see. That's a super interesting point. Yeah. So I'm going back with that. Okay. This is another question that I already have, because probably this migration and this evolution are associated with the type of antibiotic treatment that the patients are taking at the same time. That is another variable. So the question here is another popular question. Did your patient have similar antibiotic treatments? And how that is affecting the result that you are getting?
Oh, that's a really interesting question. I do not know their patient histories off the top of my head, but that is definitely something I will be looking into. Because I think, I think that definitely could have an effect on the migration, especially if you think about if they have different capacities also for like the inhaled antibiotics to get into the different regions. I think that definitely could affect how the course of the evolution. I will say they were only treated at 2 different centers, and they were both the centers were in the U.S. So we at least won't have like country to country variation in like general types of antibiotics used. But that is something I will be able to go back and look at. Thank you very much.
Yeah. So we have another question by Michaela Mosop. And she's wondering the number of migration events that were very different between the 3 subjects you showed, around 30, 100, and 230. Do you think this is only related to the Pseudomonas strain causing the infection, or could there be host-related factors involved in this?
Yeah, absolutely. I do just want to clarify at the beginning, we sequenced different numbers of isolates for the different patients, and so that is driving a little bit of what like the absolute change in number of migrations that you're seeing, but I still think your point is very good. I do think that there are differences within the patients that could be affecting the migration rate. Again, like I think probably nutrition and inflammation are going to be key factors underlying
I was wondering about that, because normally the bacteria, they will evolve to where they can obtain energy. So my question is, I was thinking about what about the environment? You know, I love metabolism. So what about the bacteria? That's my bias here. So what is it like? Is there something in that environment that is facilitating the bacteria to obtain energy, and that's why they are evolving toward that direction?
Yeah, that's a really interesting question. We're currently examining like oxytrophies to see if there are differences in the oxytrophies that the Pseudomonas have between the different regions within a given person. And so, yeah, I think we're like still in the process of doing that. But even just in the first person we've looked at, there do seem to be differences where Pseudomonas in like one region of the lung is totally heterotrophic, and then in other regions they have different oxytrophies actually. And so I'm really looking forward to finishing that to better answer your question. But yeah, I think you hit it right on the head.
Okay, so thinking about that, thinking about specific strains and how they evolve, there's an interesting question by James Gurney again. Do you think the epidemic strains like less will have more migration within a patient?
Oh, that's interesting. That's actually very interesting, because they are transmissible between patients. And so are the factors that make them transmissible between patients, does that make them more transmissible within the same lung? That's very interesting. Maybe that's why they're able to kick out the strains, like the old strains, because they can get into all the new different regions. That is very interesting.
It's an important point to follow up in the future.
Yeah, thank you.
So we have 1 minute remaining for a question. So I have one question about, you show that there is a correlation between the airway region, the damage associated to that region, and the evolution of these strains of Pseudomonas. So I know that I asked you this question before, but what is first here? What do you think is happening first? Is Pseudomonas is evolving and that is driving this immune reaction that is damaging the tissue, or is the opposite? Is first the immune response, this pressure, is inducing this evolution, or probably are both happening at the same time?
Yeah, so it's a difficult question to answer, because like we're kind of taking a snapshot in the middle of this circle. I mean, based on the arrest CF data, it seems like inflammation is occurring even before Pseudomonas has infected. And so I do think that it's going to end up being a circle where you have like some inflammation driving some amount of evolution of Pseudomonas. But then we also have examples where Pseudomonas evolves hyperinflammatory variants. And so you could then have the circle coming back, the arrow coming back, where Pseudomonas then drives inflammation in the region that it's in. And then that could potentially, further drive evolution within that region.
Yeah. Okay, Samantha, that was a great discussion. We have a lot of questions here waiting for you, but because of the time, we cannot continue. So I invite all the people who are interested about this question to contact Samantha directly. and you can find her contact in the platform. And thanks very much, Samantha. We will move to the next presentation. Okay, so the next presentation, it's Matthew Schaeffer. Dr. Schaeffer, he will discuss the interaction between Mycobacterium abscessus complex and macrophages in the CF lung, and how this is regulated by the bacterial expression of the two-component system PhoRP. So please, let's roll the video.
Hello, I am Matt Schaeffer, and thank you for attending my presentation, where I will talk about my work in understanding the two-component system PhoRP and how it regulates virulence in Mycobacterium abscessus. Before I get started, I have no disclosures. A little bit of background about the Mycobacterium abscessus complex. The abscessus complex, along with the Mycobacterium avian complex, form a group of bacteria called the non-tuberculous Mycobacterium. The abscessus complex, unlike the avian complex, are considered to be rapid growers. The members of the abscessus complex are capable of causing all sorts of infections, but the number of infections in people with CF have been increasing in recent years. These infections are often very difficult to treat because these bacteria are commonly resistant to many antibiotics. The complex is composed of 3 different species. Also, sometimes they're labeled as different subspecies, but abscessus and mycelians are the 2 most commonly seen members, with boleta occurring less frequently. Little is known about how these organisms cause infection, because there have been limited studies looking into their virulence. In the context of CF, it was originally believed that people with CF were infected with Mycobacterium abscessus that they were exposed to in the environment. Over the past couple of years, there have been a series of studies that have challenged this belief. And in these studies, they have sequenced a large number of CF isolates. They have found the presence of these groups of closely related strains of Mycobacterium abscess, which have been labeled the dominant circulating clones. And these strains are highly related and have been observed to cause infections in people with CF across the world. These studies have also identified a number of genes that have had mutations that occurred during chronic infection. This suggests that these genes may be under selective pressure, and modulation of their activity may be important for the virulence of Mycobacterium abscessus. One of these genes, PhoR, forms a 2-component system along with its partner PhoP. 2-component systems are one mechanism the bacteria use to sense their environment and modulate their gene expression to respond to that environment. Nothing is known about the PhoRP system in abscessus, but it has been studied a bit in TB. We have PhoR, which is the sensory histidine kinase, which binds to some sort of response to some sort of signal from the environment. There is no clear consensus on what that signal is in TB, but when it is exposed to that signal, the system becomes activated, and PhoR autophosphorylates, which then phosphorylates the other part of the system, PhoP, which is the response regulator. Phosphorylated PhoP can then turn on transcription of target genes. In TB, it is well known that the PhoRP system regulates many genes that are involved in virulence. It's worth pointing out that in TB, if the PhoRP system is disrupted, that those mutants are considerably less virulent in multiple infection models. It's also interesting to point out that modulation of the PhoRP system by the introduction of single amino acid changes in either PhoR or PhoP can have drastic effects in the virulence of those mutants. Looking at the abscessus PhoR protein, here I have the predicted domains of PhoR along with some of the observed mutations from these studies. They are color-coded to denote what species the mutation was observed in, and it is worth pointing out that mutations have been observed in all 3 species. Also, all of these mutations are amino acid changes, as opposed to frame shifting or stop codons, which suggests a more subtle modulation of the pathway. A subset of these mutations occur within or near the HAMP domain. HAMP domains are a sensory domain. They respond to some sort of stimuli, and that then activates them, which can then turn on the histidine kinase domain, which is adjacent to it, which can then turn on the signal transduction pathway. Also, in addition to the mutations near the HAMP domain, are a series of mutations in this region between the 2 transmembrane domains. This region has been described as a sensory loop in TB. So the location of these mutations suggests that that chronic infection may be leading to a modulation of how PhoR senses whatever the stimuli is in the environment. Looking to see what the effect of, or see how PhoRP is involved in virulence, I then created PhoRP deletion mutants in 2 different strains. Both of these strains are clinical CF isolates. One of them is a abscessus strain, the other one is a mycelium strain. To make the deletions, I deleted the PhoRP genes and replaced them with a zeomycin resistance cassette. I then did an antibiotic exclusion assay to look to see how these strains survive within macrophages. I infected THP1-derived macrophages with one of the 4 strains for 2 hours. I then treated with high-dose amikacin to kill the extracellular bacteria. I then lysed the macrophages at various time points and counted the number of intracellular bacteria. In both species, the PhoRP deletion in either the blue or purple line was significantly less able to survive within the macrophages compared to the parental strains in the solid black lines. This demonstrates that PhoRP is required for full survival within macrophages of abscessus. I then wanted to look at the role of PhoRP in an actual infection. To do this, I used the competitive index. And these experiments, you infect mice with a mix of both the mutant and the parental strain. I'm able to do this because in my case, the PhoRP deletion mutant strain is marked with a zeomycin resistance cassette. So I can plate these bacteria on media with and without zeomycin in parallel to differentiate the 2 strains. For each mouse, the CI is calculated, in which we look at the ratio of the mutant to the parental strain in the tissue compared to what was used to infect the mice with. A CI of 1 demonstrates that there is no difference between the 2 strains. A CI greater than 1 shows that the mutant strain is more fit in that infection, and a CI less than 1 demonstrates that the mutant strain is less fit. To do this experiment, I used CF mice, and the model I used was the Delta F508 strain mouse line in the FVBN background. In this model, mice that are homozygous for the mutation have the CF phenotype, and the way these mice are bred is that literomate heterozygous mice are also generated to serve as non-CF controls. Mice were infected with a mix of the PhoRP and parental strains in both of either abscessus and myceliums. I then calculated the CI of each mouse 7 days after infection. And for both species, there was a defect noted by the CI of less than 1 in both the heterozygous controls and the CF, demonstrating that PhoRP is required for full virulence within this infection model. I then wanted to see if a longer infection resulted in a greater defect. So I infected another group of mice with a mix of the PhoRP deletion along with the parental strain and calculated the CI. Here on the right, there is approximately a 10-fold reduction in the CI in both the heterozygous and the CF, suggesting that the defect seen in the PhoRP deletion mutant is much greater after a longer infection. These data demonstrate that the PhoRP system is required for full virulence in mice. I then wanted to look to see what the mechanism of how PhoRP is controlling the virulence. So to do this, I did RNA-seq of in vitro grown bacteria. I compared the gene expression profile of the PhoRP deletions in both species compared to the parental strains. I saw a fairly small number of genes that were differentially regulated, and I classified genes as differentially regulated to be at least 2-fold their difference in magnitude, along with the adjusted p-value less than 0.05. Like I said, a fairly small number of genes that were either up or down regulated. I then looked to see what were some of these genes that were differentially expressed. There was one gene, conserved hypothetical, that was the only gene that was differentially expressed in both species. There in the Mycelians strain, I also found 4 hypothetical proteins that are potentially prophage genes, along with a transporter gene that were significantly downregulated in the PhoRP deletion. In the abscessus strain, I also found a pillin and a glycosyltransferase gene that were drastically downregulated in the PhoRP deletion. It's interesting to point out that both the glycosyltransferase and the transporter gene that were identified to be differentially expressed, these genes seem to be restricted to being found only in members of the abscessus complex. They are not found in other members of the broader Mycobacterium. So it is very interesting. To wrap up my findings, the PhoRP system is required for full virulence in macrophages and within our murine pneumonia model. The PhoRP system regulates gene expression, and some of the genes that it regulates are potentially some virulence-associated genes. The next steps I'm going to take with this project is to further elucidate the PhoRP regulon, including to look at the gene expression during infection and to further describe the genes that are regulated by the system that could be responsible for the decreased virulence. I also plan to complement the PhoRP deletions with different evolved PhoRP variants. Finally, I would like to thank Greg Creeby at Boston Children's Hospitals, along with various members of the lab who've helped me along the way, Jonathan Livney at the Broad, who helped me with the RNA-seq, Patrick at Walter Reed, who has helped me with the library building and the sequencing, Lindsay Calverley at Michigan, who provided the strains, and my funding, the CF Foundation, along with Boston Children's Hospital. Finally, I'd like to thank the organizers for giving me the opportunity to talk about my project, and you, the audience, for listening. Thank you.
Thanks very much, Matthew, for that amazing talk. Super interesting about how this two-component system is regulating the capacity of the ability of Mycobacterium abscessus complex to survive within macrophages. So said that, I have one question that you said that this gene and what is reported is that if you analyze strains from CF patients, these genes gained a lot of mutations, okay, and a long time. So, and at the same time, you are showing us that this gene is important for this bacteria to survive in the host. So why this gene is being mutated if it's important for the bacteria to survive in the lung. Can you elaborate a bit more in there?
That's a great question. So it's kind of getting into kind of the second step of this process. First, I'm trying to describe what the system does, the PhoRP system. And it clearly is doing something that allows the bacteria to survive within the host and within macrophages. I'm still trying to sort out what the mechanism is. I know in TB that the PhoRP system modulates genes that are involved in the cell wall synthesis. So possibly this glycotransferase may be involved in that, where it's altering the cell wall. I'm currently pursuing that as another route. There's also this pillin that's being regulated by the system. It's very controversial role in TB, whether or not that is important. Some groups say it is, some groups say it isn't, but this is the structure. The neighboring genes of this pillin is quite a bit different. So if I had to guess, I would guess that these mutations would allow for differential expression of some of these genes, which would allow the bacteria to better survive at later points during infection.
Yeah. Do you think that there is because we know that there is a connection, as you said, between these two component system and the ability of the bacteria to build up the surface to produce different lipids. And we know that that is super important, at least for Mycobacterium tuberculosis and for Salmonella typhimurium as well, another major intracellular pathogen, to survive within phagocytes in the macrophages. Do you think that that is one of the mechanism or one of the reasons why these bacteria, there is selection for this mutation a long time to reinforce the surface and to survive within the phagocytes?
Yeah, I think that's totally plausible, and it would make sense on what's happening with patients who are infected with the bacteria. It's long-term, they're exposed to the macrophages, and they're trying to find ways to survive better, so they can do this by potentially altering their cell walls a bit.
Yeah. So we have a question here. So are there mutations in other components that correlate with the PHO mutations?
Not that I'm aware of. I don't have the full, quite as in-depth sequencing analysis of a lot of strains as I would like to have that of closely related strains. I know that there's the couple of publications from Bryant where they identified these dominating circulating clones, and they found a couple other mutations that are kind of selected for, but then they kind of circle back on these PhoRP mutations as being one of the major genes that is mutated during chronic infections and among a lot of chronic infections across the world.
Yeah, and in that setting, in the chronic infection, now what happens if we look early during the infections? Do you know what is the role of this gene in humans? You have the role in vivo, in your mice model, but is this gene being expressed more or less during an acute setting, and how that determines what is going on later?
That's a good question. I think just with the nature of the way that these infections present, it's harder to identify an early infection, but it would be interesting. I know that there's abscessus also causes multiple infections in other disease models, not just within CF. So it'd be interesting to look at some of those. It was originally defined to cause abscess infections in wounds, so that could be a slightly more easier infection model to see what role these genes play with an earlier infection, a more acute infection. But it's a different model, so it could be the PhoRP could be something limited just to the CF phenotype, or more important for a pulmonary CF infection.
Yeah, this two-component system is associated with the sensing, as you say, of the environment. The metabolic and nutritional environment around. It's not the same outside of the macrophage than inside of the macrophage, where the pH of the phagosome is extremely acidic compared to the extracellular milieu. So, do you think that this apply the function of this cluster? It will be similar in the lung compared to another tissue?
I'm not sure. I think it would be very similar to a similar environment within a macrophage, whether that macrophage is in a wound or in the lung, I would think. But it could make sense that could also allow PhoRP to mediate if it is, say, responding to pH. to be important in multiple infection models.
Mm-hmm. And following that, we have another question from the audience from Nicola Ivan Lore. How common is PhoRP mutations in CF strains? Is this mutation common in the DCCs described in CF patients?
Yes, they were common in the DCC strains. I don't have the absolute number, but I know it was one of the most commonly mutated genes among the genes that they identified. But I don't have a sense for the absolute number, but it is one of the top ones that they had identified, and we had identified in our limited sequencing, we too.
Okay, and just to wrap up, because we are running out of time, I have a last question about those genes that you saw in your genetic analysis. You have one hypothetical protein, which is super interesting. Do you have an idea of what this hypothetical protein could be doing? Is this associated with, I don't know, nutrient environment,
so there
something
was,
else?
Sorry, there was the hypothetical protein that was differentially upregulated in both species.
Yeah. Yes.
In some data since then, this gene is adjacent to the PhoRP system. And I did qRT-PCR since then, and the differential expression was not complemented. So I think it's an artifact of the mechanism I used to make the mutants. So I'm glad I didn't spend a lot of effort trying to sort that one out, but it is interesting that in looking at the TB literature, that gene is commonly differentially regulated in deletion mutants in TB mutants. So it's probably some sort of I've disrupted some regulator. So it is interesting that there was no overlap in the differentially regulated genes.
Okay, and we got a last question from the audience. Could the acidity of the CF airway help to select for Mycobacterium NTM that is better adapt to survive in the macrophages? That's like a general question.
I think it could be. The CF airways are very complex, especially in later infections, later disease states. So it's plausible that the acidity could be one of the driving forces.
Okay, well, thanks very much, Mario. That was an amazing talk. And because of the sake of the time, we will move forward to the next presentation.
Thank you.
So now we will move to our last speaker, who's Dr. Patrick Moore. He's from the University of Minnesota. And Dr. Moore will discuss with us how anaerobic organisms residing in the human lung facilitate attachment of Pseudomonas aeruginosa to the airway and epithelium. So let's move to the video, please.
I'd like to thank the organizers for allowing me to present my research at this workshop. My poster number is 414. I have no disclosures to announce for this presentation. Our previous work in our lab has demonstrated the functional capacity of anaerobic microbiota to degrade airway nuisance and to support the growth of canonical CF pathogens such as Pseudomonas aeruginosa. You can see here on this left panel, CFU counts of Pseudomonas, which was grown in the presence of anaerobes plus minimal mucin media containing Pathogenesis. This was grown over a 72-hour period, and you can see here by 48 hours, there's a 10-fold increase in CFU count of Pseudomonas in the presence of anaerobes compared to bacteria that are Pseudomonas grown in the absence of anaerobes. On the right here, you can see here this picture taken after 24 hours, this green pigment, characteristic pyocyanin pigment in co-culture tubes of Pseudomonas anaerobes. This was compared against Pseudomonas grown in the absence of anaerobes. So we wanted to check if anaerobic taxa can promote Pseudomonas airway colonization in chronic airway disease, such as cystic fibrosis. To do so, we established a co-culture system using CAL3 cells, which were grown on their airway liquid interface for 2 to 3 weeks. Once these cells are permeabilized, cell cultures were then placed in a gas permeable plate that theoretically allows for diffusion of gases through the basal artery side. These are then assembled and transferred into an anaerobic co-culture chamber. And on the apical surface, we have a mixed gas consisting of CO2, nitrogen, and hydrogen, while on the bottom we have a profusion of O2 and 5% CO2 and 21% oxygen. This allows for the oxygenation of the media and therefore keeping the viability of the cells intact. This supply of O2 and CO2 was provided by an external pump. We chose Cal3 cells because of their ability to produce excessive mucin. And you can see here, macrophage C is produced by these cells, and this is detailed by this green fluorescence. We determined that the effects of this dual oxic anoxic culture on Cal3 cellular physiology using LDH as a measure of cytotoxicity. On the right, here you can see by these blue markers, this donates the cells that are in this co-culture chamber for 24 hours. Although we see an increase in LDH after an increase in cytotoxicity after 24 hours, this was not significant. On the right here, these panels consisting of transepithelial electric resistance and echihedron, both used as proxies of cellular integrity. Although these had decreasing trends in both tear and echihedron respectively, these were not statistically significant. These data were further supported by immunofluorescence microscopy, which revealed conformal monolayers and well-defined staining of the tight junction Z01 protein that appeared as near continuous rings localized around the periphery of each cell in both the cells in the anoxic, dual oxic anoxic co-culture model and also cells growing under normoxic conditions. Finally, we used ELISA assays to quantify IL-6 and IL-8 production by Cal3 cells. Both of these cytokines showed no significant increases under the co-culture system after 24 hours, suggesting that basolateral supply of O2 and CO2 is sufficient to prevent a pro-inflammatory response. To gain a broader understanding of the physiological response of Cal3 cells to the dual oxic anoxic culture, we used RNA-seq to compare global Cal3 transcriptional gene expression to cells growing under normoxic conditions. Transcriptomic analysis revealed 148 differentially expressed transcripts, of which 170 of these were upregulated and 31 of these were downregulated. On the right here, you can see normalized counts. Angiopoietin 4 and serpinol A1 were both upregulated after 24 hours, and these are often used as markers of acute inflammation. More importantly, IF1 alpha, which is consistently expressed at low levels under normoxia, but upregulated under hypoxia and stress, also is consistent between culture conditions at 24 hours, suggesting Cal3 cells were sufficiently oxygenated. Other bioinflammatory markers were assessed, and we see that both ICAM1 and TGF-beta1 were the only ones upregulated after 24 hours. Finally, since we would eventually use the dual oxic anoxic culture model to assess bacterial colonization of the apical mucus layer, we compared mucin-related gene expression between conditions. Among detectable transcripts, which are listed below here, no significant differences were observed. These data demonstrate that the dual oxic anoxic culture of Cal3 cells yields minimal changes in host cell physiology. So in order to see if anaerobic microbiota can colonize the apical surface, we used a defined anaerobic bacterial consortium derived from airway mucus from an individual with chronic sinusitis. This consortium was grown in mineral mucin media as a carbon source, and we chose this for its dominant bacterial taxa, including Vionella, Streptococcus, and also Protella species. Cal3 cells were apically challenged for 24 hours and imaged using scanning electron microscopy. You can see here by these crudely imaged bacteria that the airway microbiota, the anaerobic microbiota, was able to colonize the surface of the Cal3 cells. Importantly, anaerobes were recovered via PBS wash, and also cells lyse with Triton X100, which suggests that both anaerobes are able to proliferate or survive on the apical surface and also attach to the surface of the Cal3 cells and potentially invade the cells. Recovery of Protella species was noted using a Prevotella selected media under anaerobic conditions, which confirmed that the apical oxygen concentrations were sufficiently reduced to facilitate strict anaerobic growth. On the right here, you can see cytotoxicity. And although ABC, or the Anaerobic Bacterial Consortium, caused an increase in LDH release or cytotoxicity, this was not statistically significant compared to cells grown under normoxic conditions. We then used RNA-seq to profile the Cal-3 transcriptional response to anaerobic challenge. Contrary to our expectation, only 5 genes were differentially expressed relative to untreated controls. However, when we looked at normalized counts, we do see an upregulation of mainly pro-inflammatory cytokines, and this would suggest that anaerobic microbiota induces a pro-inflammatory response after 24 hours in this dual-oxic anoxic culture model. In support of downstream pathogen colonization experiments, we use fast protein liquid chromatography, or FPLC, to determine whether anaerobic challenge alters Cal-3 mucin integrity relative to unchallenged cells. To do so, we challenged cells for 24 hours. We purified the mucin, and then we used size exclusion chromatography to assay their integrity. As expected, this chromatograms revealed 2 distinctive peaks: peak 1, which is mainly higher molecular weight mucins, and peak 2, which is mainly lower molecular weight mucins. Treated cells revealed that there was a significant reduction in peak 2, which would suggest that anaerobes can degrade low molecular weight mucins compared to higher molecular weight mucins. And on the right here, these are just area, these are units of area under the curve. You can see here, peak 1, there's no statistical difference, whereas peak 2, there's a significant decrease of low molecular weight mucins following anaerobic microbiota treatment. So, given that anaerobic consortium used in our model can both elicit a pro-inflammatory response and alter mucin integrity, we hypothesized that the anaerobic microbiota can enhance Pseudomonas colonization of Cal-3 cells. To do so, we pre-treated our cells with anaerobes and anaerobic consortium. These are 2 individual different experiments. We then washed, after 24 hours in the dual-oxic anoxic culture model, we then washed the cells to remove any unbound or residual anaerobes and treated further with Pseudomonas aeruginosa for 2 hours. We then lysed the cells and we quantified Pseudomonas colonization of Cal-3 cells using colony forming units. So you can see here on the left, when we washed after 24 hours and infected with Pseudomonas, you can see here a significant increase in P. aeruginosa CFU recovered from cells that were pre-treated with anaerobic bacterial consortium. To further demonstrate that the enhanced pathogen colonization was due to mucin degradation and not another unidentified process, mucins isolated from Cal-3 cells treated with the anaerobic bacterial consortium and untreated controls were used to coat a microtiter plate, followed by the addition of PA14 for 2 hours. As expected, mucin coating of untreated mucin led to a 1.5 log reduction in bacterial attachment relative to PBS control. Consistent with our other assays in this dual-oxic anoxic culture model, ABC, or anaerobic bacterial consortium treated mucins resulted in a significant increase in P. aeruginosa binding to the microtiter plate compared to untreated mucins. So to confirm that our mucin purification process, such as the use of guanine hydrochloride, did not affect P. aeruginosa viability, plates were also coated with mucins degraded with human neutrophil assays and isolated using the same process. Neutrophil assay treated mucins resulted in a similar PA14 attachment to BBS controls, confirming that anaerobic microbiota can enhance pathogenic colonization of a mucin-coated interface. Finally, to assess the contribution of individual anaerobes to Pseudomonas aeruginosa colonization, we challenged COL3 cells with representative isolates of 3 of the most abundant genera in the anaerobic consortium, as I listed earlier. Again, these consist of Prevotella, Streptococcus, and Veillonella. So these were challenged for 24 hours, washed, and then subsequently infected with P. aeruginosa for a further 2 hours. Interestingly, contrary to studies, Streptococcus did not have an effect on P. aeruginosa colonization, even though this is a known mucolytic bacteria, whereas a Prevotella species was able to significantly increase P. aeruginosa colony-forming units prior to after treatment with Prevotella species. Veillonella did not have had no significant difference on P. aeruginosa CFU counts. These data demonstrate that while anaerobic microbiota of the respiratory tract facilitate enhanced colonization of the epithelium by airway pathogens, it is clear that they do so in a species-specific manner. So my conclusion is that dual oxic anoxic cold culture enables extended direct study of anaerobic host interactions at the airway surface, in which anaerobic microbiota stimulate a pro-inflammatory response while also degrading mucin integrity at the epithelial interface. They stimulate the anaerobic microbiota stimulate the colonization of the epithelial interface by canonical pathogens, and more importantly, this study or this research paves the way for future studies, allowing us to shed light on potential pathogenic mechanisms of anaerobic airway microbiota in disease progression. I'd like to thank everyone in the Hunter Lab, specifically Talia and Sabrina and Leslie, who have helped with the RNA-seq, FBLC, and scanning electron microscopy. Thank you.
Okay, Patrick, thanks very much for that super brilliant presentation. Super interesting, the model that you established, and that led us to understand a bit more how the anaerobes interact with all the pathogens in the CF airway. So we have a couple of questions from the audience, and also I have another questions. I was wondering about that reduction in the peak that you showed about the mucins. So what are the byproducts that are being produced by these anaerobes, and how these byproducts are increasing the capacity of Pseudomonas to attach to the surface? Can you elaborate a bit more on that, please?
So we think the main byproducts being released from anaerobic fermentation is small chain fatty acids. We fully haven't characterized what small molecular weight mucins have been released in that second peak, so we don't know what simple sugars or core 1, core 2 types of mucins have been released. But we think that would allow for further binding of Pseudomonas to the surface of cells and to mucins, to the core backbone of mucins.
Okay, so based on that, we have all the questions from the audience. We have another question from Joshua Chandler asking, are you measuring release LDH in the apical environment, basolateral media, or both?
Just apical environment. Then we haven't looked at the basolateral side, and that's a great question. We just focused on the apical side because that's where the anaerobic environment is. But it would be a great question to answer, is that to look at what LDH is being released into the basolateral side in a more aerobic environment, or the environment that has most CO2 and O2 diffusing through.
Great. As this is a new model that you are sharing with us, there are some questions about if you can describe the experimental setup in more detail, please, just to give some.
Yeah. So again, I grow my cells for 2 to 3 weeks under airway liquid interface. We then have this manifold that we bought from Koi Labs that was previously used in a gastro cell line. We have permeable membranes that sit on top of the manifold. We have pre-made gaskets. The 24-well trans will sit in those gaskets, and then we transfer that into the anaerobic chamber. And then we have two lines that go in, one internal, one external, for diffusion of CO2 and O2. And we use 5% CO2 and 21% oxygen because they're the standard incubator conditions that we grew the cells in, and we wanted to keep with that for our 24-hour to 72-hour experiments. This protocol is further demonstrated in my bioarchive paper, and I'm happy to, if that person wants to reach out to me, email me, and I can send them that paper, but it's on bioarchive at the moment.
Excellent for that, for sharing that information. We have more questions for the audience. We have one from Pradeep Singh, and he's saying, very interesting work, Patrick. I realize this is a challenging experiment, but have you examined whether anaerobes increase Pseudomonas growth in sputum from subjects that are not yet infected with Pseudomonas?
Oh, we haven't done that. Can you repeat the second part of that question?
Yeah. Have you examined whether anaerobes increase Pseudomonas growth in sputum from subjects not yet infected with Pseudomonas?
Not to my knowledge. We have only done it in porcine gastric mucus. So I guess we haven't done it in sputum itself, but only in porcine gastric mucus. I think that's being currently done at the moment.
Okay. So with that, we have another question said, does EPS laid down by the anaerobes alter the Pseudomonas binding rather than the degradation?
True, it can, but this is one, because we have shown previously that anaerobes support Pseudomonas growth, and one way, this is just one mechanism of how the possibility of anaerobes influence Pseudomonas infection, or colonization, I should say. So I'm sure there's other methods, as I've shown through the RNA-seq. It produces a pro-inflammatory environment, which it also can aid in Pseudomonas colonization. But this is just one mechanism of which anaerobes can support Pseudomonas colonization in the CF airway or in any respiratory airway. So I'm sure there's other factors involved, but this is just the one mechanism that we've figured out, or one mechanism that's potentially aiding in Pseudomonas association.
Yeah. So the other something that you share with us is that there's one specific anaerobe, Prevotella, that is promoting this attachment of Pseudomonas. What is the difference between Prevotella with the other anaerobes? What are these bacteria doing?
So I think with Prevotella, it's compared to, this was such a surprising result as Streptococcus can degrade mucins. We've shown in the lab that Prevotella can degrade porcine gastric mucin somewhat, so it's not surprising that it can increase Pseudomonas colonization. I need to do more further experiments with that on its own, how it's degrading Cal3 mucin after infection, or after treating the cells with Prevotella. So I think I need to delve into that a tiny bit more going forward.
Excellent. So we have another question. Great talk, Patrick. This question by Jennifer Bomberger. In addition to the epithelial attachment phenotype that you assessed, does the anoxic environment with other microbes impact Pseudomonas aggregate growth in the mucus layer above the epithelium?
I'm not sure. I'm not sure. I've shown previously back in my PhD where mucus can obviously cause aggregation. So I don't know if the breaking down in the mucus increases aggregation after Pseudomonas. Potentially it does, because you get more potential access to the mucin core backbone, and also access to the surface of the cell, which can increase aggregation and also potentially invasion. I'm not too sure. That's a more confocal question. Microscopy question, and that'd be a great thing to do. Cal3s are somewhat hard to sometimes hard to use on confocal, but that's a great question.
Okay, and here there's one question that also I had, but Jose Antonio Barros Martin, he is asking, you showed that anaerobes induce inflammation.
Mm-hmm.
Have you checked if in your model there are specific anaerobes more pro-inflammatory than others?
Yeah, we haven't elucidated that yet. That's another thing we need to go over. Is to show, say, Prevotella, as you were talking about before, if that's inducing more of the pro-inflammatory environment compared to other anaerobes. So that's the thing that we need to tease out going forward. This was just more to show that, this is a potential mechanism of how the anaerobic community within the CF airway can promote Pseudomonas and potentially other canonical CF pathogens in increasing colonization, increasing infectivity of those canonical CF pathogens.
Excellent.
That's a great question. We need to tease out which, because we do show in our 16A sequencing that there is quite a few anaerobes in there that it would be great to tease out which ones are producing that pro-inflammatory environment and also causing that change in the mucin integrity after treatment. Sorry.
Excellent. Yeah, that's a super important point. So Patrick, thanks very much for your time presented with us. That was a brilliant work.
Thank you.
So now we will move to the general session, the final part of this symposium. So we have some minutes remaining. So I would like to know if the speaker, they want to share something extra with us, if there is any other idea they would like to tell us. If there are no other ideas, other things to say, well, I would like to, I don't know if Matthew can, Matthew Wargo, the other chair in this workshop, would you like to add anything before leaving?
No, I think we're good.
Excellent. That was a super interesting workshop, all the different variables that are influencing in the capacity of different pathogens to attach and to infect the CF airway. I hope that we can have more of this in the next year as well. So with that said, I would like to thank the presenters for their time and effort for sharing with us their valuable work. If you have any remaining question, of course, please don't hesitate contacting the speakers. Their email and contact are available in the NACFC platform. And of course, as well, with Dr. Matthew Wargo, we're happy to have served as chair in this workshop. And we are looking forward to answer any questions that you may still have. With that said, we close our workshop now, and I hope to see you again soon. Have a nice day.